Waters. And before I start, I just want to say my presentation is very different. I'll be focusing on the analytical tool that we use for glycan characterization, specifically for therapeutic proteins. As some of you probably know, that Waters is a tool provider. We develop analytical instrumentations and in the area of liquid chromatography, mass spectrometry, and informatics. So the glycosylation is a key quality attribute, and based on the International Conference on Harmonization guideline, it requires for analysis of carbohydrate content, structure profile, and the characterization of glycosylation site and the polypeptide chains. And there's a variety of therapeutic proteins that are glycosylated. Actually, probably about 70% of them are glycosylated that's currently on the market. And the glycosylation can be very simple, like on the monoclonal antibody. This trastuzumab is an example. It only has one N-glycosylation site, and the profile of glycan is relatively simple. And or you can have somewhat very complicated glycosylation on a protein such as EPO. It has 3 N-glycosylation sites and 1 O-glycosylation site. And half of the protein mass are contributed from carbohydrates. And another protein here is etanercept. That's one of the most difficult, challenging protein that I ever worked with. It's got 13 O-glycosylation site and 3 N-glycosylation site, and then they call that protein a snowflake protein because not all 2 molecules are the same because of the complexity and the heterogeneity of glycosylation. And from my group, we do a lot of method development, focusing on glycoprotein characterization, and then look at the sugar. So there's a there's a multi-strategy in terms of characterizing glycoprotein, and we can look at released glycan, which will be the focus of my presentation, or look at the peptide, glycopeptide, looking at the site heterogeneity. And then sometimes we do middle up, middle down experiment, look at subset of the protein, like say for monoclonal antibody, it will be light chain, heavy chain, or FC and FAB domain. And we'll always do inter-protein analysis, just look at the general profile, looking at the deconvoluted mass spectrum. And then last but not least, looking at the monosaccharide analysis. So for today, I'll be focusing on release glycan analysis. And then I've done a variety of analysis, and the carbohydrate glycan analysis is probably on the top of my list as far as difficulty, because when we look at sugar, you're looking at composition, connectivity, and the configuration differences. So it's a lot more difficult just if I compare it to primary amino acid sequence of a protein. So this is very, there's a lot of structural isomer in terms of carbohydrates. And here's the class of N-glycans. We have high mannose type, complex type, and hybrid type. I like to use the Oxford nomenclature because I saw, George, you have that yesterday on your slide. So those are the 3 main type of N-glycans. And also, why is it so important looking at glycosylation for therapeutic proteins? Because glycosylation has a huge impact on the drug's efficacy and safety. These are just some of the highlights, and I'm not going to go through every one of them. Just maybe a list, like say, for example, salicylic acid content. It has a huge effect on the half-life time of proteins such as EPO, and also alpha-gal. It can be very immunogenic to patients. Here's the conventional way of doing glycan profile for therapeutic proteins. And majority of our peers from a biopharmaceutical company laboratory, that's the methodology they use. And so, for example, so they will start from a protein, they do PNG-SCF deglycosylation. Usually it's overnight or at least several hours. Then once the glycan's released, they will label it with a fluorescent tag. I would say maybe 60 to 70% of lab uses 2-aminobenzoamide, and then the rest of them split between 2-aminobenzoic acid and other labeling techniques. And then once the glycan's labeled, then it goes through purification, and then follow with either LC optical analysis or CE optical analysis. And for our group, we tend to do using optical and MS all combined in an integrated system. So you get fluorescent signal and a mass spec signal for confirmation purpose. And the overall workflow using the conventional method would take about 1 to 1.5 days. The most time-consuming part comes from protein deglycosylation step using PNG-SCF, and then the labeling usually takes somewhere between 1 to 3 hours, and the drying step. So altogether, it takes 1.5 days, which is pretty long, and it's error-prone because you have so many steps involved. Now, so what's new from Waters? To address those challenging aspects of the sample preparation, and also, I forgot to mention, using 2-aminobenzoamide, you get good fluorescent signal, but you get very poor mass spec signal. I don't know if any of you have experience using 2-aminobenzoamide. So from Waters, because of those challenges, we developed a new labeling strategy using a novel compound called RapidFluor MS reagent. So if you look at the structure of this labeling reagent, it contains 3 functional groups. So if I go from left to right, so this is the part that does the reaction with glycans once it's released from protein. And this is a part that has a strong fluorescence signal. Okay, so this is a quinone fluorophore. At the end is triethylamine moiety that it gives very high MS signal when you do electrospray ionization. So then this part does the rapid labeling. You have strong fluorescent signal after labeling and a strong MS signal. So here's a more of a detailed structure, and it shows you the RapidFluor MS label react with glycan when it's released from glycoprotein under physiological pH and slightly alkaline pH. You will get glycan in a glucosamine form. So the reaction happens, we say 5 minutes, but it's actually seconds. So then the sugar and the labeling compound form very stable urea linkage. And now we go from the conventional labeling method and all the way to 30-minute labeling method, taking advantage of this rapid labeling reagent. And also we collaborated with New England Biolab, and for the deglycosylation step, they have this called rapid PNG-SCF component, so that we were all incorporated into the whole workflow. So with the rapid PNG-SCF, we can do the deglycosylation step in less than 15 minutes. That's why overall reaction is under 30 minutes. And then here's the comparison of using conventional labeling strategy versus the new label. And this is a base peak intensity mass spectrum chromatogram. And the top is, this is the same sample. This is NIST monoclonal antibody standard. And the top chromatogram is the 2AB labeled from 10 micrograms of starting material. And at the bottom is the new labeling reagent, RapidFluor MS, starting from 0.2 micrograms of monoclonal antibody. So if you correlate the signal, you get over 100 times more sensitivity improvement in from electrospray ionization, which is a huge deal. Because of that extra sensitivity, you can now able to see the low abundant glycan in the sample. And this just to show that when we mix glycan from IgG and the fatuing and using the new label, if I just to overlay the fluorescence and base peak intensity mass spec, you can see that this label works well for neutral glycan and small biantennary glycan all the way to highly silated glycan from the fatuing. And then you can see the mass spec data and the fluorescence data correlates very well. And here's to show the benefit of using RapidFluor to enhance the sensitivity. So this is, again, fluorescence chromatogram. And you can see that there's some low intensity peak in the early eluting retention time. If I zoom 20 times, oh, the animation didn't work. So here. Okay, so if I zoom into this region, you can clearly see there are a lot of peak in the early eluting chromatographic time, and those are real glycans. So that with this new label, you now can see this very well, versus if you use conventional method, those peaks probably doesn't stand out. You might see a little bit in fluorescent, but you're never going to see a real signal from the mass spec. And then here's just to show what it looks like for low abundant glycans. This is again the human IgG glycan. Top is fluorescence, bottom is BPI-MS, and you can see this low intensity biantennary singly silated glycan right here. It matches the mass spec chromatogram matches with the fluorescence. And if I sum the scan here, you can see their majority of the components here are multiply charged with a little bit salt adduct. Okay. And you can do MS-MS for this low abundant glycan. And here's the annotated MS-MS fermentation for the biantennary afucosylated singly silated glycan. The fermentation pattern is analogous to 2-aminobenzoamide labeled glycan. You predominantly get glycosidic bond cleavage, and it gives you a very clean serial neutral losses. Then you can annotate the structure using the fragmentation data. And here's another example on the alpha-gal containing glycans, how this new label can help identify the alpha-gal containing glycan. So this is mouse IgG glycans. And then here, you can see, again, fluorescence, BPI-MS. And there's the peak right here. There's a shoulder peak right next to the biantennary fucosylated glycan. And originally, I find out that the main peak and the shoulder peak has the same exact mass. So what is the shoulder peak? Oh, I did MS on both main and the shoulder peak. You can see that the fermentation data looks very different from these two peaks. And I conclude that the shoulder peak is actually alpha-gal containing glycan, and the main peak is not the alpha-gal containing peak. The reason why I see a marker fragmentation ion that only come from the alpha-gal containing, but not this one, is because the loss of gal, and I'm sorry, it's a loss of glycan, because it's coming either from here to here. If you have galactose attached to you have two individual terminal galactose, you're not going to see the loss of glycan. The only time you're going to see it is when you have an alpha-gal structure. It's just very structure informative. Now I'm going to switch gear. I talked about this labeling, the utility from using a high MS signal fluorescent label. Now, what about the analytical workflow that supports this new label? And here's we call biopharmaceutical scientific platform from Waters. It comprises the front-end sample preparation for doing the glycan sample prep, and then you have ultra pressure liquid chromatography and benchtop time-of-flight mass spectrometry, and then controlled by informatics software called Unify. And it does a variety of analysis, but one of them is designed for glycan analysis. We call glycan application solution. And inside there's two main workflow. One is called glucose unit plus accurate mass, and then the other one is DDA, fermentation. So the glucose accurate mass, I know you probably never heard about it. This is to because we use liquid chromatography for separation, so the retention time from the chromatography actually can be used for glycan confirmation. So the glucose unit is really the calibrated glycan retention time using a dextran ladder, and then combined with the accurate mass from the mass spec, you use these two information, and you use a pre-populated scientific library that has all those information for individual glycan, so you can do a search. So this is one of the workflow. The other one is DDA. You depend on fermentation spectron, and then you search using a SynGlycan software from Premier Biosoft, which I'll show you what it looks like. So the GU, the glucose unit value, I mentioned, I'm not going to go into detail because I don't have a lot of time today. It's really a calibrated liquid chromatography retention time for sugar using a dextran ladder. Okay, it can normalize the retention time across different instrumentation, different user, or on different days. So it's a very useful little tool. So the scientific library I mentioned is that we actually had ongoing collaboration with Professor Pauline Rudd in Ireland. Her group does a lot of database for glycans. One of them is taking advantage of the RapidFluor MS fluorescent tag, and then they create a scientific database for each glycan. In that database, you have a glucose unit value coming from the liquid chromatography, and then we have a mass information altogether. So if you just see an example of what the scientific library looks like for each individual glycan component, you have the structure, you have the labeling reagent. Here is selected for RapidFluor MS, and you have experimentally determined the glucose unit value, and with the standard deviation. Here's the mass with the label, and here's item tag, meaning where does that glycan come from. There's a host of protein that has this sugar structure. Okay, so this is what the experimental data looks like if you incorporate this workflow using the calibrated retention time and accurate mass. So this is the dextran ladder. So you run that, you get a calibration curve, and the calibration curve will be automatically applied to any subsequent unknown sample. Then each peak will have a glucose unit value calculated from this calibration curve, and then you have the mass chromatogram. So for each glycan peak, you have the retention time value from the curve, and you have the extracted mass information from your mass spec. And together, you get automated assignment.
2 minutes.
Oh, okay, so here's what it looks like with assignment. You have the library search results with the structure annotated. You have the mass information and the data table. All right, so quickly go to the DDA. Basically, you can collect MS fermentation automatically, and you can export process the data into a SynGlycan software in a variety of file format. And here's just an example to show that I actually identified this glycan from my previous workflow. I know it has alpha-gal containing structure and NEUGC containing saccharide, so it's a immunogenic glycan basically. So using both workflow, I was able to ID this glycan. So we are currently still developing a scientific library for this new labeling reagent, and we hope to apply this label for more complex sample, a more high complex glycosylated proteins. And here's the people that I want to acknowledge. Matt Lauber is our chemistry expert from Waters. I worked a lot with him on developing this new reagent. And Paula is from New England Biolab. She's the one developed the method for rapid PNG-SDF release. And then also I'd like to acknowledge Professor Pauline Rudd and her group for the collaboration. And I thank you for your attention. I'm more than happy to answer your questions.
Thank you. Let's thank you. Question down the front here.
Very nice presentation, but the question I have here is, how do you use this in a clinic? In particular, when you're looking at the glycan profiling in patients that have a particular disease compared to normal population. I can see the beauty of that, but there's a problem.
Yes, that's a very good question because for now, our group is, if you don't know, our group is called Pharmaceutical Lab. So we developed this tool specifically for glycan profiling used in like quality control and just the whole biopharm arena. And we have a separate group that's more life science, omics focused. And I was hoping to extend this technology to their group. Maybe they can work on the clinical diagnostic tool with this labeling reagent. The benefit of this, I know from collaboration with Pauline, she has done a lot of work for biomarker, looking at 2AB labeled sugar from, like, say, healthy patients versus disease, and then they can compare the profile of the fluorescence and then see what are the glycans being changed, what's new, what's the concentration has been changed from those two different samples. So I think definitely there's utility. I mean, I just hoping that somebody will pick it up and do more work.
Just to add, we were doing this kind of work several years ago, in collaboration with Roland Contreras at Ghent University, where he was looking at these glycan profiling, especially expressed on different receptors. The problem we confronted is the purity of the protein. And so I could see here, maybe if you're doing this profiling on antibodies, maybe taken from monoclonal antibodies, which is highly purified, how does that relate to the natural antibodies in terms of this library that you're generating?
The library we generate will have to start with very pure protein.
How do you get that? That's the problem.
But those are, yeah, well, you can do gel purification, then it's still not pure enough. Yeah, but yeah, that's a very good question. I know that the part of the protein release and the populating the library is done.
Okay, perhaps we can discuss this further over lunch, because I think we're running short on time here. So next speaker, please. And thank you again to Dr. Yu.